Erwinas Neheris.
Okay, can you hear me? Yes? Yeah. Well, it's a pleasure to be here. Almost as cold at home in Göttingen. I was asked to say a few words about myself before starting the talk. So, would call me a biophysicist working at the Max Planck Institute for Biophysical Chemistry in Göttingen, Germany. I'm now working on synaptic transmission, more specifically on the release of mechanisms of release of neurotransmitters from synapses. For some time I've worked on the release of catecholamine from adrenal gland. Release of hormones is very similar to release of neurotransmitters. But this is not the topic of my talk today. The topic of my talk today is ion channels, the field which Bert Sackmann and myself got the Nobel Prize in 1991. So, in a way, it's a historical talk, unlike the previous talk, which was a fascinating example. Yeah, please. Okay. So is this better now? Yeah. Okay. Now I can speak very softly, I think. Yeah. And you still hear me. Very good. Okay. So I just want to say that you just heard a very fascinating talk about very recent technology. My talk will be about technology we developed some 30, 40 years ago. And what I think I want to convey to you is how much can change within this time period, within the time period of, say, the lifetime or the active lifetime of a scientist. So, to start on the more historical track, let me go back quite a bit, namely to the origins of our understanding of bioelectricity, the fact that there is electricity in our body. And this, of course, starts with the work of the famous Italian scientists Luigi Galvani and Alessandro Volta, who about 200 years ago found out that there is electricity in living tissue, namely by showing, this is the work of Galvani, that you can make the muscle of a frog twitch when you stimulate the nerve by an electric shock. 100 years later, the famous Spanish neuroanatomist Ramón y Cajal showed us in his beautiful drawings that our nervous system is made up of this filigrane network of structures, which we now call neurons. And we now know that the brain is a network of about 10 to the 12 of such neurons, which are connected with each other via synapses, each cell making on average contact to about 10,000 other cells. When you look closely into the drawings of Ramón y Cajal, you see in many places these little arrows, you know, and this is what Ramón y Cajal thought, found out about the signal flow in the nervous system. And we can admit that he was mostly right, although all his conclusions were derived from looking through the microscope onto dead and fixed tissue. Of course, at that time, he didn't know exactly what these signals were, which he was postulating. However, around the same time, in 1868, Julius Bernstein, a physiologist in Berlin, built this complex machine which allowed him to record very rapid small electrical changes at the cut end of muscle and nerve, And indeed what he found was this small negative deviation in the electrical signal, which was only about 1,000th of a second long duration, and which basically was the first recording of what we now call the action potential or the nerve impulse. He also developed his membrane theory in 1902, which says that the signals, the electrical signals, are generated at the membrane. Each cell is surrounded by a membrane which separates electrically the inside, which is a conducting medium, a fluid with ions, from the outside, which is again a conducting medium, the membrane forming a kind of insulator. But Bernstein's hypothesis was that the signals are generated across this insulator, like in a cable where the signals are generated between two conductors separated by an insulator. Okay, the big advance in our knowledge about the electrical signals, the action potential, came when British physiologists Huxley and Hodgkin, working on the squid, found out basically what happens, how a nerve impulse is generated. The squid has this very big axon, a nerve fiber, which is basically a membrane cylinder, several centimeters long and almost a millimeter thick, where you can induce axial wires longitudinally and can apply voltages across the membrane and can measure current. And they succeeded in finding that when you depolarize the membrane, if you apply a step-like positive potential to the membrane, you can measure currents which consist of one component carried by sodium ions, a transient inward current followed by a component which is carried by potassium ion, an outward current. And what they could show is that they put the kinetics of these currents into differential equations and solved the differential equation by mathematical computation. At that time, not with a supercomputer, but by hand with a multiplying and adding machine. They could demonstrate that a membrane cylinder with these properties will generate a wave-like signal which propagates along the axon. So, from this work, it was known that the action potentials, the nerve impulse, is a consequence of permeability changes in the nerve membrane. The big question of that time was, what's the mechanism of these permeability changes? Why does the membrane suddenly become permeable for ions, and this in an ion-specific way? This was a kind of questions which fascinated me already as a high school student. I was interested as a high school student in biological things, in plants and animals, but I was also interested in technical devices, in radios, in clocks, and so on. And the fact that there is bioelectricity in electricity in the body was something very fascinating. So I decided that I want to know more about this already as a high school student, and that I should become a biophysicist. Now, at that time, it was not possible to study biophysics like some of you do. You either had to study biology or physics or chemistry, was strict separation in the classical disciplines. So I decided to study physics, but then switch into biology as fast as possible. So I was very lucky that I entered the laboratory of Dieter Lux after returning from a period that I studied in the United States with a Fulbright fellowship. So I came back to Munich and was looking for a PhD project which would allow me to study bioelectricity. Okay, so Dieter Lux was running a laboratory at the Max Planck Institute for Psychiatry in Munich, and he offered me a PhD project working on electrical excitability of neurons. The neurons were not in the squid, they were not in human, they were not in mice or rats. The neurons which we studied were in snails. At that time, physiologists looked for the biggest structures that were available for studying a certain problem, and snails were found to have very large cell bodies, spherical structures of a few 100 micrometers sometimes, and you could put microelectrodes into them, record the voltage between inside and outside, and this is actually what you see here, recording from my thesis, where I did this. I stuck electrode into the cell. I measured a resting potential of about minus 70 millivolts, which is typical for a neuron. Then what you can do with these classical microelectrodes, you can inject current through the electrode and depolarize the membrane. And if you do this at relatively small strength and stop depolarizing, the membrane returns back according to the membrane time constant. But if you stimulate stronger and surpass a certain threshold, then this happens, what was shown by Hodgkin-Huxley, namely an influx of sodium makes the inside membrane potential more positive, followed by the outflux of potassium, which brings the membrane potential back again. So it's the typical phenomenon, one second, I think a nice example of just the nerve impulse. Now, as said here, the big question was, what is a mechanism? There were many ideas about this mechanism around in the literature in the 1970s and 60s. Of course, Hodgkin-Huxley had cast their equations into their results into equations, assuming gates which open and close. This suggested the concept of ion channels, pores in the membrane, which would open and close. However, there were other ideas. Some people argued that these conductances are so ion-specific that just a pore couldn't be so ion-specific. They hypothesized that this current should be mediated by ion carriers, because at that time, only ion carriers were known to be very ion-specific, like valinomycin, which is a very specific ion carrier carrying one potassium ion at the time across the membrane. So my friend at the time, Bert Sackmann, and myself decided that we wanted to prove the concept of ion channel, Hodgkin-Huxley concept, by showing that by demonstrating that we can record step-like changes in current when these pores open and close. Now, the problem was we had to expect that these currents were very, very small. And in fact, some indirect measurements suggested that the currents should be on the order of magnitude of a picoamp. Now, picoamp is 10 to the minus 12 amps. This arrow here shows you the kind of order of magnitudes of currents. You know, 1 ampere is a current in light bulb. 1,000th of that, a milliampere, is a typical current in electronic circuit. This is also the magnitude of currents that you can measure in the biggest biological structures, the squid axon, but typical currents in a biological system are still a factor of 1,000 smaller. Microamp, these are the currents here which are elicited by the nerve when the nerve signals to the muscle that it should contract, initiating contraction. We now know that the neuromuscular transmission is mediated by the neurotransmitter acetylcholine, which is released from the nerve ending and acts on the postsynaptic membrane, opening ion channels. This neurotransmitter is stored in little vesicles inside the nerve ending, which are released. So we know that the end plate current, the total signal generated by the nerve, is made up of so-called miniature currents, which are the currents which are generated by a single such vesicle containing acetylcholine liberating. And now we know that even these miniature currents are made up of ion channels, still a factor of 1,000 smaller, 10 to the minus 12 amperes. The problem was that all methods available at that time, which would allow to measure current in a biological preparation, had an intrinsic noise of about 1/10 or 2/10 of a nanoamp, 100 times larger than the signal we wanted to measure. So we knew we had to come up with a new method to develop something drastically more sensitive. And what we came up is now known as the patch clamp technique. In the patch clamp technique, you do not push microelectrodes into cells, but you place a somewhat bigger electrode. All these electrodes are glass pipettes with very small opening. You place this pipette touchingly onto the surface, trying to isolate this little patch of membrane for the electrical measurement. I mean, the pipette has fluid inside. It's connected through a electrode, through a silver wire to an amplifier. And the idea was, here magnified, that if we were lucky to isolate this patch of membrane, if there would be an ion channel on the patch, if we apply the voltage across the membrane and measure the current, that we should be able to measure this current in much higher sensitivity, and if we are lucky, see the opening and closing step-like changes when ion channels open and close. And this actually happened. We succeeded. Now, these very first measurements were done on muscle membrane, where we know near the place where the nerve had been touching the muscle. Bert Sackmann had gotten experience in enzymatic treatment of a preparation in removing the nerve so that we could place our pipettes onto the site where normally the neurotransmitter acts on the muscle. We had, of course, very small amount, tiny concentration of acetylcholine, the neuromuscular transmitter, in the pipette. And when we adjusted the acetylcholine concentration properly, we could see these step-like changes in current. Now, what turned out is that actually the key to this measurement, it's relatively simple, you know, but the key to the measurement was to obtain a very good seal, a tight contact between the muscle and the membrane, between the pipette and the membrane. And the importance of this seal comes from a very simple equation which you can find in electronics textbooks, namely the minimum noise that you can obtain in an electrical measurement is given by the so-called Johnson noise, the resistor noise. The effect resulting from the fact that ion electric charges are discrete, electrons or ions, you know, and so by thermal agitation, this discreteness of ion of charges creates this Johnson noise. Can you see the arrow? No. The arrow is not showing up? No? Yeah, well, so I was trying to use my arrow here because it shows up in both, whereas the laser shows up only here. Okay, anyway, I'll use the laser from now on. I'm sorry. So the Johnson noise, which is given by the square root of the thermodynamic constants, kT, times the bandwidth of the measurement, divided by the resistor value, or more accurately, the impedance of the signal source. So you can solve this equation for the resistor required to obtain a noise small enough, you know, at a certain bandwidth. And if you do this, you can find out that you need an impedance of the signal source at least several hundred megohm in order to obtain a noise of sub-picoamp in the bandwidth of a kilohertz. So we knew that we had to work on this, and we kept working, improving the seal, and after some time we actually succeeded to get really good seal and then to obtain very well resolved step-like changes when we recorded from these structures, namely the membrane of the muscle, which contained acetylcholine receptors. Now, at this time, then, biochemists had advanced to the point where they could isolate from electric organ so-called acetylcholine receptors, membrane proteins with 5 subunits, which 2 subunits have acetylcholine binding sites. And what obviously happens is, when these binding sites are occupied by acetylcholine, then the membrane, the protein undergoes a conformational change, which opens a passageway for ions. So what we see here is basically, in real time, the conformational changes of a single protein molecule. Now, with the improved resolution, we could go back to the question which originally fascinated me, namely the electrical excitability. And this then was done by a postdoc in the lab, Fred Sigworth, who was able to record from electrically excitable muscle once more such currents, depolarizing the muscle membrane with a step-like voltage step, like Hodgkin-Huxley. He saw these individual negative blips, short episodes of inward current. When he then formed ensemble average, which means just averaging many of such traces, he got a phasic inward current, very much like Hodgkin-Huxley had shown in the squid giants. So with these 2 types of measurements, we had to prove that both electrical excitation and chemical excitability is mediated by discretely switching ion channels. So this was basically what we wanted to achieve. And so far I've reported about our own work. What I will try to do in the rest of this lecture is to show what type of consequences this discovery had, what type of consequences the availability of a technique had, which is 100 times more sensitive than the previous ones. And this will not be our own work, but summarizing work of many laboratories worldwide who have used our technique to study ion or currents in biological tissue. And this resulted in a number of surprises. The first surprise was that ion channels are not restricted to so-called excitable tissue, but that they fulfill a large number of tasks in basically all cell types of our body. Of course, they are there in excitable tissue, but they are also found in kidney cells, where they regulate fluid secretion, in liver cells, red blood cells, even in bone cells, and particularly also in the cells of the sensory organs, where they mediate the conversion of physical quantities like light or sound or chemical things into electrical signals. So while we thought originally that ion channels are restricted to electric excitable tissues, and that maybe 5 or 10, or if it comes high, 15 different types of ion channels might be found in biology, we now know that there are about 200 different types of ion channels fulfilling all these different tasks. Let me give you a few examples. So this is an example from the heart, where an electrical signal, very much like a nerve action potential, generates the contraction of the heart, but the action potential in the heart has a very long shoulder. Its upstroke is carried by sodium ions, like in the nerve, but it has a very long shoulder for the muscle, heart muscle, to be able to contract, and then there is this return to baseline. And in order to form this shape in the ventricle, you need at least 4 different types of potassium currents, which switch on each of them in with a certain time course, switch on and off. Now this is true for the atrium, for the ventricle. In the atrium, where the contraction is being generated, the action potential is quite different, with the initial peak followed by a lower shoulder. And to generate this kind of signal, you need at least 8 potassium channels. And of course, a number of more channels for generating the inflow, like sodium channels, calcium-dependent channels, channels depending on hormones to regulate the heartbeat by the autonomous nervous system, and so on. So this was a big surprise, so many channels. Second surprise was that ion channels are targets for pharmaca. Now, here is one example of a so-called action of a so-called calcium antagonist, a class of substances which had been used clinically to lower blood pressure, and who had been postulated that it acts through reducing the calcium influx into the heart. Now, this experiment conducted by Harald Reuter, a colleague from Bern, showed that in the cardiomyocytes there are voltage-dependent channels, very much like sodium channels, which, however, are permeable for calcium ions. So the medium influx of calcium, which then initiates the contraction of the muscle. And he was lucky a few times to have a patch where there was just one single channel of a certain type, namely this voltage-dependent calcium-carrying current. And when he depolarized the membrane, he saw that this current, this channel, was rapidly flickering open-close. If he did the same experiment under the influence of this calcium antagonist, the number of openings, the frequency of opening was much lower. The channel was reluctant to open. Consequently, the net influx was smaller, contraction was weaker. Okay, showing in a clear way the function of this drug. Okay, So it turned out that channels are unique drug targets for various reasons. And let me just give you two further examples of the action of drugs onto channels. There is this substance memantine, which is the second most prescribed drug for ameliorating the symptoms of Alzheimer's disease. It's not a cure for Alzheimer's disease, but it helps a certain fraction of patients to cope much better with the symptoms. It turned out to be a blocker of a certain type of glutamate channels in the brain. You may know that glutamate is the most important excitatory transmitter at synapses in the brain. And one subtype of these channels is blocked by the substance memantine. And as the people who developed this drug, who brought it to the point where it is a FDA-approved drug, assured me that patch measurements were very important for characterizing the drug's action. Another example of a drug acting on a channel is Kalydeco, which again is an FDA-approved drug for cystic fibrosis. Cystic fibrosis is a disease where a mutation in a chloride transporter, in a chloride channel, leads to perturbed fluid secretion. And it's a very serious condition, particularly for children who very often die at a very young age. And this drug boosts the action of this transporter, of this channel, if the mutation leads to reduce all cystic fibrosis patients, because the majority of mutations in this channel prevent the channel to reach the plasma membrane. Most mutations in this channel lead to the degradation of the protein already in the endoplasmatic reticulum. But those patients who have a mutation which allows the protein to reach the plasma membrane, for those, this Kalydeco is a drug which allows them to lead a normal life. So it's a example for a so-called genotype-specific medicine for personalized medicine. And it brings us to the next surprise, namely that defects in ion channels cause diseases. And I have here just a few of them listed. In particular, many arrhythmias of the heart are due to changes in ion channels, myotonia, certain types of epilepsies, type 2 diabetes is a very prominent example, cystic fibrosis. So this list could have more than 100 entries, because there's really a huge number of diseases known to be related to mutations in ion channels. And this is not surprising, given the multiplicity of ion channels. I told you that we thought there might be 10, 15 ion types of channels. Now we know that the genome has about 200 genes which code for ion channels. Each of these points here, endpoints, representing one of the sequences which code for ion channels. You see there are potassium channels, there are ligand-gated channels, there are, well, all kinds of channels, many of them which have appeared or been characterized only very recently. Let me go into one more example for a so-called channelopathy, a pathology related to ion channels. And for this, I come back to the heart, with the action potential with a long shoulder. And one of the very common cardiological problems is the so-called long QT syndrome, a cardiological finding which can be recognized in the electrocardiogram, where the action potential is prolonged. This shoulder phase is prolonged, leading to all kinds of complications in the generation of the heart rhythm. And this syndrome, namely lengthening of this action potential, of course, can come about in two ways. Because, I mean, the repolarization happens at a time point when the influx through sodium current and calcium current is balanced by the outflux through potassium, through outward currents. So whenever the outward currents become bigger than the inward currents, this repolarization will happen. Now, this balance, or the shift in this balance, can come in two ways. Either by a gain of function in a sodium channel, namely delayed inactivation. In some patients, it has been found that this specific type of channel has a mutation so that there is a long tail of inward current which prolongs shifts the point of repolarization to longer times. But likewise, it can come about by a loss of function in a potassium channel. And again, many patients have been diagnosed as having a mutation in this KCNQ1 channel. And just like this loss of inward current, a higher outward current can lead to the same phenomenon, namely a delayed repolarization. So there is a total of 12 different LQTS syndromes which are known, which can be differentiated, and 8 of these are linked to ion channel genes. Okay, so another example relating to pharmacology, namely about drug safety. It turned out that a large number of substances which had been developed as drugs for very different purposes failed in the clinical tests due to side effects on the heart. And it turned out that one particular channel, the so-called hERG channel, is particularly attractive or sticky for molecules which otherwise would have properties which makes them as good drugs. And it turned out that many of these substances influence the activity of the hERG channel and lead to arrhythmias. So in order to avoid this, the FDA ruled already more than 10 years ago that all candidate molecules, drug for any target, have to be tested for possible side effects on the hERG channel before a substance can go into clinical trial. And a new initiative, again by the FDA, called SIPA, is preparing now protocols for extensive testing of drugs for their influence on human cardiomyocytes, because it had been turned out in between that the side effects of drugs on heart go much beyond those on the hERG channel. So these were two examples for channelopathies and for drugs. Let me close with another example of a somewhat surprising role for ion channels which is a little bit amusing, I hope, namely the role of ion channels in the sensation of heat and cold. You probably know from your physiology or biology lectures that in the skin there are multiple sensory organs for touch, for pain, for itch, and also for heat and cold. And it turned out that in these nerve endings there are specialized ion channels which open and close in a temperature-dependent fashion. All these channels are of a certain type of a class of channels, namely the so-called TRP channels, channels which serve many, many different functions. Among them, these heat-sensitive channels, they all have the name TRP with a letter and something. TRP because the first member of this class of channels was found in the photoreceptors of insects, where the channel produces a so-called transient receptor potential, a part of the light response of the insect photoreceptor. Now, as I said, starting from this first member of the this class of channels, it's found that this is a very big class of ion channels. In fact, I saw there is one poster out there about some of these TRP channels. So, if you want to know more, that there are some TRP channels which open in the very cold, others which open only at noxious heat, and still some more which open and close at intermediate temperatures, and together they cover the whole range of physiological temperatures. Now, I promised a surprise. What is the surprise? The surprise came when researchers found out that these channels not only are sensitive to temperature, but are also influenced by chemicals. That certain chemicals shift their open-close behavior. And now, if you look what these substances are, you can see that capsaicin, the extract from red pepper, not surprisingly, activates the heat-sensitive channel, the channel which ordinarily senses heat in our body, you know, and the extract from mint, the menthol, opens cold-sensitive channels. So, with this knowledge, you now know better why you feel hot when you eat a hot meal and why you feel cold when you drink a cup of mint tea. Thank you for attention. Well, most important slide. Okay. Of course, I mentioned that the original discovery was together with Bert Sackmann. At the time when we made the most advances in the technology was when we had postdocs in the lab. Fred Sigwarts, I mentioned his contribution, who is now a professor at Yale. Alain Marty, now professor in Paris, and Owen Hamill in Galveston, Texas. And let me mention once more, all of the second half of my talk was referring to literature data obtained by hundreds of colleagues all over the world. So, thank you.
Thank you. And right away, do we have any questions? Shall we do?
So you covered a lot on channels of let's say mammalians or humans, but still looking evolutionary. I mean, for example, all life evolved from the single organism, you know, like archaeans or bacteria, you know, and then we getting to the organisms with billion cells. So that means the single cell which develops to the organisms, it has to sense also you know, it could first have to be this mechano sensation also, that it senses also the environment. So can you elaborate a little bit on these channels and even evolution, you know, from these primitive organisms, you know, that it has to be big shift, you know, towards something, you know, where you have billion cells?
Yeah. Well, I mean, even bacteria and viruses have channels, you know, where channels, say, serve for volume changes, you know, to maintain the volume. Single cell organisms have channels which do all kinds of things, which sense. I mean, light sensing is typically done through, of course, a second messenger cascade, where at the end cyclic JMP in our photoreceptors is opening opening channels. The development goes from, I think, originally potassium and chloride channels through mechanosensitive channels, as you mentioned. There is a class of channels called the piezo channels, which are very important in development, in formation of tissues, you know. Sodium channels, which we were interested originally, come very late in development. Basically, I mean, the voltage-dependent sodium channels, which mediate action potentials, became only important when organisms became big, so that rapid signal transfer was necessary. Calcium channels play important roles very early for all kinds of coupling of electrical changes to biochemistry, because, of course, calcium influx triggers a huge number of calcium-dependent processes in the cell. So, I think that was my answer here to your question.
Thank you.
Thank you very much for this exciting talk. I would like to ask you, the most focus is always held on the exciting cells and the roles of those channels in the exciting cells, like nerves or cardiac cells. What do you think are the major roles of the same channels in the cells, in the dormant cells, or not exciting, like chondrocyte in the cartilage or osteocyte in the bone, that they for the sensing of mechanics, or what is the role of them, do you see?
Yeah, well, I mean, of course, the electrically excitable channels are important for, as you mentioned, or as I explained, for action potential, for contraction. The mechanosensitive channels are important, first of all, for sensing mechanical stress, then for osmotic regulation, and in particular for they mediate signals important for tissue development, you know. I mentioned that calcium permeable channels are important for all kinds of links between electrical phenomena and biochemistry, you know, changing intracellular calcium, leading to contraction, leading to phosphorylation, dephosphorylation. So, I mean, I should emphasize that channels are not everything. Channels are part of a huge regulatory network which is established in our cells, you know, but they have their role in this regulatory network next to second messengers, next to receptors, G protein coupled receptors, you know. Yeah, that's it.
One more here quickly.
Sorry.
I talked about the evolution of the channels, but what we have now in the environment, for example, we basically are now facing some technological breakthroughs. You know, we have telephones, we have internet, we have even Wi-Fi, you know, around us. So do you think these things like Wi-Fi would influence also our channels or even evolution of our channels? You know, how that actually influences our body, you know, and perception of these things which we cannot measure actually, but they're existing, and how, in terms of the channels, you know, do they really sensing all these things, you know, which are creating extra, you know, with the technological breakthroughs? Yeah.
Well, I mean, of course, Wi-Fi influences our measurements. You know, we have to be very careful to screen your apparatus from these influences. Wi-Fi and also just a 50-cycle hum, but there is no reliable evidence that it would influence the workings of individual cells. First of all, because cells are very small, you know, so any gradients, electrical gradients, have to be very big in order to have a strong influence. And then all this happens inside conducting media, which is a shield, you know, to such influences. And as I said, I mean, I don't know of any reliable evidence that say normal exposure would have a big influence. There was a question up there.
I have a bit practical question. How did you ensure that during each experiment the same amount of ion channels are used? Thank you.
Maybe I gave a wrong impression. First of all, we talk about single channel recording. In fact, usually during a measurement, there are quite a number of channels on a given patch, you know. But say for our first measurements, for the case of acetylcholine activated channels, we just adjusted the concentration of acetylcholine so low that of channels would be a rare event, that we saw either nothing or maybe one channel or a maximum maybe 2 or 3 responses superimposed, you know. So the example I showed about this cardiac channel of Harald Reuter was a kind of a special case where actually due to the behavior of the up and downs and the lack of superposition of events, Harald Reuter could be sure that he had only a single channel, you know. Now, I think the question, your question included, how do we know that it's always the same kind of channel? Is this what you wanted to know? Well, we can't guarantee. I mean, we have to say the experiment with the acetylcholine channel, we have to compare measurements with acetylcholine in the pipette versus measurements without acetylcholine. And even without acetylcholine, we now we often see some kind of step-like changes, you know, mostly then of different amplitude, differently influenced by changes in voltage. But the most important criterion, of course, is that the channel you want to study is influenced by your experimental parameter, in that case, the concentration of acetylcholine.
Yeah, so from the point of material science, nanochannels are like nanopores, basically. And so there is a big variety of these ion channels as you described. So what are the physical dimensions of the inner diameter as compared to these ions and perhaps other biomolecules, so we can use them like for other purposes?
Well, I mean, this relates to the actual ion transport. And there is now crystal structures of many different types of ion channels. The most known one, I mean, the very first ion channels which have been resolved by X-ray crystallography are potassium channels, which have a kind of internal pore, which is just big enough, at least at the narrowest point, to let a potassium ion pass, in the sense that the hydration of the potassium ion has to be replaced by coordination with carbonyl groups of the protein backbone, you know. So there actually needs to be a tight fit in order to have ion selectivity, you know. Other channels which are not ion specific have somewhat larger channel structure, but that's very different between different types of channels.
I would like to ask about the... I'm here.
Where? Here. Yes, okay.
I would like to ask about the possible future of patch clamp, especially regarding automated patch clamp techniques. Yeah. Because, well, some techniques are being developed that allow scientists to patch multiple cells at the same time, and, well, no longer very experienced patch clampers may be needed, so, well, that's okay, but I would like to ask, what do you think, will the automated patch clamp will become the norm of?
Well, it depends what kind of studies. Actually, there are two challenges to patch clamp. One is automated patch clamp, the other one is optogenetics, you know. Regarding automated patch clamp, of course, this is important when it comes to mass screening, to drug screening, you know. However, I think the current status is that automated patch clamp works with a number of cell lines, you know. I mean, in automated patch clamp, you don't put a pipette onto the cell surface, but you have a substrate with nanopores, with little holes, where from a cell suspension you suck a cell onto this hole, you know, and then something happens similar to the contact with the glass, that the seal forms, and that you actually can record currents on this nanopore on a flat substance. And this can be parallelized so that you can do many studies simultaneously. This works with certain cell lines, which have to be very pure, you know, which have to have certain surface properties, but it's good for drug screening, but it doesn't work, say, for cells in tissue, you know, or if you want to study a channel in some type of cell, you would have to invest quite a lot of work to create a cell line which has this channel with this property. So I think from that point of view, for academic research working on specific problems, the conventional patch clamp still has a future. In particular, for the study of these many mutations which have been diagnosed in patients with channelopathies. Now, the other thing is optogenetics, which promises to provide fantastic possibilities to both influence the behavior of cells and measure responses by purely optical technology. The basis of optogenetics, on the one hand, are so-called channel rhodopsins, rhodopsin-like molecules, which form a channel which is light activated, and if this channel is expressed, this channel rhodopsin is expressed in a given cell type, you can depolarize the cell and generate action potentials just by shining light onto them. Now, you can combine this with genetically encoded probes, optical probes, for either calcium changes or else for voltage changes, and so that you ideally can both measure changes in voltage optically as well as inducing changes, you know, and this kind of technology more or less fulfills the dreams of neuroscientists who I mean, the dream is to be able to see online, you know, well resolved, the activity of various neurons, and at the same time being able to influence their behavior, all optically. Now, this is definitely a technique which is on the rise, which will replace lots of pendulum studies, you know, but still, I think the resolution is not as good, at least with certain aspects. And it still, of course, may have side effects which are not known so well. I mean, in electrophysiology, we know the devil. We know what the problems are, you know. And I think many researchers using optogenetics will still have to find where the devil is, you know. But it's definitely a technique of the future.
Thank you. I think we have time for one more question, please.
Maybe you can also comment how the numbers of channels are regulated in the cells. Is that like constantly the permanent number of channels in the cell throughout its life? Or when you use blockers, some of them get blocked, but maybe new channels are opened immediately, or maybe some hormones regulate that there are more of channels opened, more of channels synthesized in the cell. How is that regulated, the numbers of the channels?
Yeah. No, I mean, of course, this is a very dynamic process. You mentioned many of these possibilities, and as a rule, I would say that the number of channels is regulated just like the expression of any other molecule is regulated according to a developmental program or to the demand, you know. So there's nothing special about regulation of ion channels. Yeah, well, I mean, usually blockade of channels is a transient phenomenon. Of course, I mean, if you say block acetylcholine channels with bungarotoxin, you know, which is a permanent, more or less a permanent block, the muscle develops something which is called denervation hypersensitivity. Also, if you cut the nerve, you have on the muscle this denervation hypersensitivity, in the sense that somehow the muscle feels that it doesn't get input anymore and expresses new channels, you know. That's just one example of a kind of response. But as I said, everything which is found in other types of proteins also applies to channels.
Thank you. Now we have to move on, so thank you once more for the answers. Thank you for the questions, obviously. And now, for the school students that are leaving, I would ask them once more to leave through the top of